Hey, Mr. P here. So we are into DNA structure, and so before we talk about the structure of the entire DNA molecule, we need to spend some time today talking about how DNA structures itself at the smaller level, meaning the monomer level. We need to talk about nucleic acids in the sense of repeated nucleotides, and we're going to spend a little bit of time talking about how DNA, from a chromosomal standpoint, protects itself and what the repercussions are if the molecule doesn't protect itself. So, what is nucleic acid? So we've obviously heard nucleic acid before. When we talked about organic chemistry, or the organic molecules, carbohydrates, proteins, nucleic acids, and lipids, nucleic acid was one of those major divisions of organic molecules. And so we learned a while back that nucleotides are the molecules that are in charge of storing information. Nucleic acids can be in the form of DNA and RNA. Okay, DNA, remember, is deoxyribonucleic acid, DNA, and RNA is ribonucleic acid, and so we have RNA. So these are the names, these are how they get their names. DNA and RNA are for short. One of the molecules that we've talked about a lot is adenosine triphosphate, and it's really easy to forget that it belongs to the class or the division of molecules called nucleic acids, but as we've seen before, it contains the same three components that all nucleotides contain. In fact, there is a ribose sugar, adenine, which is a nitrogenous base, and there are phosphate groups. And when we talk about, on the next slide, the components of a nucleic acid or a nucleotide, it is going to contain everything you see here. It's going to contain one phosphate, one sugar, one pentose sugar, just like ribose is here, and one nitrogenous base. We definitely want to include ATP into the discussion of nucleic acids, but for the purpose of this chapter or this section of the syllabus, we're going to kind of focus our attention on DNA and RNA. But just remember, adenine or adenosine triphosphate needs to be kept in the same group of organic molecules called nucleic acids. Okay, so when I say nucleotide, what am I talking about? I am talking about the building blocks of nucleic acid. And so at the basic level, any nucleic acid, both RNA and DNA, are both repeated nucleotides. And so one nucleotide contains a phosphate group, a sugar, which can either be deoxyribose or ribose, but both of which are 5-carbon sugars and are in the class of 5-carbon sugars known as pentose, and they contain a nitrogenous base. Now there are 4 DNA nitrogenous bases: adenine, thymine, guanine, cytosine are all DNA, okay, or all constitute or make up DNA molecules, and then uracil is kind of the odd one out. And RNA specifically is made up of uracil. Now, uracil is going to take the place of thymine, and so RNA is going to be composed of adenine, guanine, cytosine, and uracil, while DNA is going to be composed of adenine, thymine, guanine, and cytosine. So, all nucleic acids are composed of nucleotides that are composed of any 4 nitrogenous bases, depending on the type of nucleic acid we're talking about. But, again, as I said at the beginning of this video, I wanted to talk about the structure of the nucleotide. You can see that there is, in the middle of this nucleotide, and by nucleotide I mean this entire molecule over here to the right, the center of this molecule is a pentose sugar. Okay, a pentose is a 5-carbon sugar, and before we start kind of radiating out of the center, I want to talk specifically about what the carbons are for and what the carbons do. And so we can't really see the carbons in this picture, but every time we have an angle in this pentose, you can assume that there's a carbon. And so we have the first prime carbon here, the second prime, third prime carbon, fourth prime carbon, and then fifth prime carbon. The top of this pentose is an oxygen, and you can see that here. Okay, so we have 1, 2, 3, 4, and 5. That's where we get our pentose. The 1 prime, or the first prime carbon here, is attached to our nitrogenous base, and in this case it's adenine. It could have been cytosine, it could have been guanine, and it could have been thymine if this particular nucleotide is a DNA nucleotide. The fifth prime carbon, which we see here, fifth prime carbon is attached to the phosphate, and phosphate, as we've talked about in previous chapters, is a PO4. Okay, so you can see P with 4 oxygens attached to it. This is our phosphate molecule. Now, when we talk about how these nucleotides kind of structure themselves, the phosphate and sugar are going to make up the sugar-phosphate backbone. Okay? Sugar-phosphate backbone. And the nitrogenous base, which is here, is basically where all of the information for the particular DNA molecule is going to be kept. So, backbone, phosphate, and sugar, and then nitrogenous base is going to obviously contain all of the information, and that information is in the form of C, G, T, and A if you're RNA, or if you're DNA. Okay? So, how are these nucleotides going to go together? Well, I think the next logical thing to talk about is the division between purines and pyrimidines. A and G, if you notice here, and they are obviously labeled with their name above their ring structure. Adenine and guanine are both double ring structures, and when I say double ring, I mean 1, 2, 1, 2. And if you notice, thymine and cytosine are both single ring structures, so they only have one ring. So what is the purpose of having double ringed bases and single ring bases? Well, we need to get into base pairing rules. Base pair rules. So what are the base pair rules? Well, the base pair rules are A pairs with T and C pairs with G. Always. Okay? When we are talking about DNA, we are always going to use these base pair rules. A pairs with T and C pairs with G. Now you'll also notice that I wrote 2 lines between A and T, and I wrote 3 lines between C and G. These are the bonding, and I should say hydrogen bonds, okay, that hold this strand and this strand together. Now if you notice, A and C, or I should say A and G, so this is a double ring base, this is a double ring base, this is a single ring base, and this one is a single ring base. Notice that we always pair a double ring with a single ring in both cases. So we're always going to pair A and T. So A is going to pair with T, and C and G are going to pair together. And that's to keep a uniform width or a uniform thickness to our DNA molecule, which you'll see in a later slide. But for now, adenine and guanine are both the purines, and T and C are both the pyrimidines. Okay, so it's kind of tricky at times to remember purines and pyrimidines. I mean, they both start with a P, they both end in ES, they both sound very similar to each other, but purines, the way I remember it, because we're here in Kansas, is that Kansas is pure ag. Okay, what is the largest industry in Kansas? It's agriculture, and because you can remember ag, A and G, being basically the abbreviation for agriculture, and that agriculture is the largest industry in Kansas, you can remember that because Kansas is pure agriculture, A and G are the largest bases. Okay, so ag, A and G basically are the abbreviations for the largest bases, and then by default T and C make up the smaller bases. Okay, and so remember when I talked about we wanted to make sure we had a uniform width or width. Think about this. If we paired a single ring base, and I know squares are not the actual symbol or not the actual shape, but if we put 2 squares, 2 single ring bases together, and then right below it in this DNA molecule we put 2 double ring bases together, and then bonded those together, our DNA molecule would bulge out and then would pinch back in if we had, say, this, okay, where we had a single ring and single ring, double ring, single and double like it should be, this is the one that is correct, and then we pinch back in with a single and single, our DNA molecule would basically be really wavy and would look really, really odd. The fact that we have a single and double always means that we will always have a very uniform width throughout the entire length of our DNA molecule. Okay, so now I'm zooming out a little bit further, so notice that we started this discussion talking about the actual nucleotide, which you can see here. Remember, we have a phosphate, a pentose, and a nitrogenous base. Okay, so this whole thing right here would be a nucleotide. Notice too, like we talked about, we have a pyrimidine, a single ring base, bonded to a purine. And remember, purine, A and G make up our purines. It is a double ringed base, guanine and adenine, two ring base. Our pyrimidine is thymine and cytosine, just a single ringed base. We have a single and double bonded together, double and single. That helps to maintain this uniform width as we go through this DNA molecule. But now we've zoomed out and we're not looking at or not focusing on the individual single nucleotide. We're focusing on a series of basically two nucleotides that are bound to the complementary nucleotides on the adjacent strand. This brings up another concept I want to talk about. I've talked originally about hydrogen bonding, so these are the hydrogen bonds. And between T and A, there are two hydrogen bonds. And between G and C, there are three hydrogen bonds. And the way I remember that is C and G and 3 all rhyme. C and G equal 3. T and A equal 2. Okay, so adenine and thymine are going to bond together twice. Adenine or cytosine and guanine are going to bond together 3 times. So those are the hydrogen bonds that are going to basically hold the two strands together. Remember from our previous discussion of bonding that the hydrogen bonds that can be seen right here are the weakest, and so just like a Ziploc bag is held together by the zipping mechanism, these hydrogen bonds are going to be very analogous to that zipping mechanism and allow the Ziploc bag to be easily opened and shut. This DNA molecule wants to basically not separate itself here in the backbone. It only wants to separate itself here through the center of this DNA molecule so that it can expose the bases, but it doesn't actually ever detach the nucleotides from the actual strand of DNA. But let's kind of get a clean slate here and focus on this DNA molecule without all of this bonding talk. So remember, we have a thymine and adenine bonded together. Thymine and adenine bonded together. Remember, it bonds 2 times. Guanine, cytosine, remember, bond 3 times. We also have a pyrimidine bound to a purine, and down here we have a purine bound to a pyrimidine. But let's just look at this strand. This nucleotide, and again we're focusing on this nucleotide up here. Remember I said this was the 1' carbon, 2, 3, 4, and 5th prime carbon. This one down here, 1', 2', 3', 4', 5'. Now we need to talk about how nucleotides actually bond to the next nucleotide. And that's going to bring up these numbers up here. And I know it's kind of hard to see because all of these kind of run together, but this right here, this is referring to the 5th prime or the 5' end of this DNA molecule. And down here, 3', this is the 3' end of this molecule. And so if we look at the DNA molecule as a whole, it's running 5' to 3' in this direction. Okay, 5' to 3' in this direction. And while that isn't so important now, it's really, really important when we get to DNA replication, which is the focus of the next chapter or the next video. On this side, 5' is down here, 3' is up here, and so this DNA molecule is running in a 5' to 3' in the up direction. Okay, so 3' here, 5', we are running 5 to 3 up. So over here, on the left side, we're running down. On the right side, we're running up. And that's the first time in this video we start to realize that the DNA is antiparallel and that it is essentially going to fold itself into a double helix. One of the reasons it can fold itself into a double helix and the fact that it is antiparallel is because both sides of the molecule are running in opposite directions. And that's going to be important when we talk a little bit later. Okay, but for now, we have a 5' end, we have a 3' end. We are bound purine to pyrimidine, A to T, C to G, always. T and A are bound twice. G and C are bound 3 times. The 5' carbon in every single one of these nucleotides is always attached to the phosphate. Okay, so the phosphate is attached to the 5' carbon. The adjacent nucleotide's phosphate is attached to the 3' carbon. And so when we build these DNA molecules, we are always going to build in a 5' to 3' direction. Why? Because we are always adding the next nucleotide to the 3' side. So where's the next nucleotide going to attach itself? It's going to attach itself here to the 3' end, the 3' end of this nucleotide, okay, or the 3' end of this nucleic acid. Over here, they are flipped, so notice that now this is the 5', 4', 5, 3', 2', 1'. And so, again, the phosphate of its own nucleotide is attached to the 5', just like it is over here. And the 3' is where we're going to attach our next phosphate when we join the next nucleotide. Okay, this is zooming out a little bit more. Now we're not focusing on 4 nucleotides, we're focusing on essentially 8 nucleotides. Again, we still see this uniform width that is maintained by bonding a purine, which can win adenine. Okay, we can bond adenine to a thymine. We can bond guanine to a cytosine. Guanine, cytosine, thymine, adenine. And so this is our DNA molecule. Again, C and G always pair together, A and T always pair together. It's a nice uniform width because we are maintaining the 1-ring and 2-ring bonding. A and T, remember, bond twice. C and G bond 3 times. Twice, 3 times. Hopefully you see some kind of unifying themes as we keep zooming out of this DNA molecule. But notice that the backbone is composed of a phosphate or an alternating phosphate pentose, or sugar in this case, deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose. On the other side, we have deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, and a phosphate. Okay? So, these bonds right here, okay, the bonds that hold the phosphate and sugar together are considered phosphodiester bonds. So the phosphodiester bonds can be seen there. They are incredibly strong. They are a form of covalent bonds, and they are the strong bonds that hold the backbone together. The individual nucleotides, and remember, this is a nucleotide, phosphate, sugar, base, is bound to the next nucleotide by this phosphodiester bond, and we would have linked those 2 monomers together using a hydrolysis reaction known as dehydration synthesis. I know you've heard that term before. Dehydration synthesis is basically removing water, so we would have pulled out an H2O and linked those together, creating a sugar phosphate backbone that is hydrophilic. Remember, hydrophilic is water loving, which is a good thing because this DNA molecule is actually out in the nucleus, or it's within the nucleus. Also, basically any water that's composed or is part of the fluid or the cytosol that makes up the interior of a cell doesn't actually do anything to the backbone because it is hydrophilic. The nitrogenous bases are basically very reactive and so need to be protected inside of this backbone, which they are really well because of the strength of these phosphodiester bonds. Again, the two strands of the two outside backbones are held together by the hydrogen bonds. The hydrogen bonds holding the two strands together, and that produces an antiparallel structure of this DNA molecule. Again, we have a 5' end and a 3' end, so this molecule is running this way, and we have down here 5' and 3', so this right side is moving that way. Because we have two rays running in opposite directions, that's where we get this antiparallel. Okay, so in this section of the syllabus, one of the skills that IB requires you to know how to do is drawing a simple diagram of the structure of DNA. And so when you do that, I would always encourage you just to start with one nucleotide. And the nucleotide, as we've talked, has just a sugar, right? And on that sugar is a phosphate and a nitrogenous base. In this case, let's say it's a T. Remember, thymine is a single ringed base, and so therefore need to be shown with a single ring structure. This, remember, is the 1, 2, 3, 4, 5th prime carbon. Next to this nucleotide, okay, we are going to attach the next nucleotide, which we're going to attach this phosphate. Off of it, we're going to produce its sugar, and then maybe this one is a double ring, which we can call A. On this one, next phosphate, maybe it's a single ring, which is C, because remember, A and G are the double ringed structures. This is all we would need to have on this left side. Remember that this strand is running 5' to 3' down, because 5 to 3, if we physically draw a line from our 5 to our 3 carbon, we are going from top to bottom, just like we are on this left side. Now, T, remember I said, is going to bond twice to A, and A is a double ring structure. A is going to bond twice to T, and C is going to bond 3 times to G. Okay, so here's our hydrogen bonds. These bonds right here or right here are the phosphodiester bonds. Okay, they're the bonds that hold our backbone together. And on these, okay, you can see that this would be an adequate drawing of basically a series of 3 nucleotides on the left and 3 nucleotides on the right. Notice that this one is running 5' to 3'. Notice on our left, our houses or little pentose are facing up, and on this side, our pentose are facing down. Okay, this would be a drawing. Everything is labeled. This one would have to be a T, right? C pairs with G with 3 bonds. A pairs with T with 2 bonds, and T pairs with A on here with 2 bonds as well. Okay, that would be an adequate drawing to get all your points for drawing a simple diagram of DNA. Okay, so I think that's where we're going to end it for this video. In the next video, we're going to talk about how DNA molecules actually structure themselves to be supercoiled. We've talked about everything you need to know from a structural standpoint in terms of how a nucleotide comes together with other nucleotides to produce the actual ladder that is a DNA molecule. But now, once we have the DNA molecule or the actual nucleic acid produced, we need to talk about how the DNA molecule needs to be supercoiled in order to fit it inside of the confined space of the nucleus and keep it protected as the cell goes through its normal cellular activities and how it replicates DNA. But for now, see ya.